Hey all, a very warm welcome on this channel. This is Dr. Jyoti Bala. In this session, we are going to discuss about significant prediction tools, especially related to protein secondary structure prediction. So if you are working in chemistry, biotechnology, drug designing, bioinformatics, this session will be important for you. Or maybe you want to explore such areas for your different projects. So do check out this video. I will be showing you 3 of the best freely available tools along with the practical tutorial and demo. Let's get started. As we all know, protein is a very important macromolecules having diverse functions in your cell signaling, biological processes, antigen antibody interaction, drug designing, diagnostic, and so on. So if you are working in plant sector, microbiology, biomedical area, or human biology, you have to deal with protein, its structures, its sequence, its interaction, its functionality. So for a different project, you require to understand protein. And here we know that the function of these macromolecules comes from the sequence only, because these primary sequence information is responsible for certain structure in terms of 2-dimensional, 3-dimensional, and later on the higher structures, and based on that they interact with different molecules and they are having some function. So if you want to explore protein in terms of functionality, we have to get the ideas about their sequences, also their structures, both at secondary, tertiary, and later on at the higher structure and interaction level also. So here in this session we are mainly focusing about the secondary structure. In my previous tutorial I've already demonstrated how you can get the sequence information from UniProt, NCBI databases if you will go to NCBI protein section. So either you can go to NCBI or UniProt, you can get the primary sequences. If you are working in new protein or de novo, then you will get it from sequencing. So in any way, we get the sequence information first, we analyze the sequences and we get the ideas based on the sequence also, and later on based on these sequences there are computational tools and platform which can predict the secondary structure and tertiary structures. So in this session we will be focusing about the secondary structures, which are the best tools which are freely available, and if you're new also, how you can start working on structure biology and computational biology of protein part. And in my upcoming session I will be demonstrating you how you can do the tertiary structure prediction and modeling work also along with the refinement. So if you have missed watching the UniProt analysis or NCBI, the other bioinformatics fundamental tutorials, I request you to watch the bioinformatics playlist and do check out those tutorial also. So let's start with the 2-dimensional prediction. So the initial step will be to get the sequence information. If you are handling de novo, then you can perform the protein sequencing work and you first get the amino acid details about your peptide and protein, and based on that you can perform your secondary structure prediction. Otherwise, if you are working on known protein or maybe variant protein, mutant protein, in that cases we can go to certain databases and from there we can get the sequence information. So either you can go to NCBI or you can go to UniProt, and there are many other platforms also from where we can get the sequences of our protein. So first thing will be to get the sequences for our secondary structure predictions. After that, if you are a beginner, I wanted to give you 3 tools which are freely available on easy to use and validated and cited one also, so that you can use them for your thesis, report, and for your actual projects. So in this session we are going to discuss about CyPred, SOPMA platform, and JPred. The SOPMA platform, if someone is beginner and if you haven't used SOPMA platform earlier, then kindly use this URL and come to this page. You just have to provide the primary sequence, the sequence information about your peptide or your protein, and based on that this platform is going to predict the secondary structures. Similarly, this is CyPred it is very cited and very useful platform. It has multiple other features also. So not only you can predict the secondary structures, like in secondary structures of your protein has different forms like helix, turns, coil, and this overall gives the functions of the protein. So in CyPred we have multiple different kind of features also. So not only the secondary structure prediction, but high-end secondary structure predictions, the analysis, if you want to get the ideas whether your protein is having different kind of polarity, atomicity, then whether they have some region which is in the extracellular region or transmembrane, all these prediction this platform would do for you. So just check out this platform also. This is the URL. By using this URL you can come to this platform. Then we have JPred. JPred is also a very useful cited platform. Here not only your secondary structure but other information like Ramachandran plot, ligand interactions, tertiary structures, a lot of different information related to protein, if that protein is having some role in pathways, enzymatic activities. It is integrated with other platforms, so you can download the files, the informations. But in this session we will be focusing only for the secondary structure prediction part. So how you can get the secondary structure information, or how you can either predict the secondary structure. So you can use the JPred also. Let's do some demo. I will be using the same BCL2 protein which we have used for our UniProt analysis, and with that same protein let's do some SOPMA, CyPred and JPred analysis, so you will get much better understanding about these platforms. So let's get started. This one is SOPMA platform. You can use this platform for the secondary structure prediction of your peptide and protein. So if you have samples related to your protein, you require sequence information. So the sequence information in my earlier videos I've already shown you. Either you can get it from the UniProt or maybe from NCBI protein databases, or if you are handling de novo or new protein, in that case you might be having your own protein sequence sample. So whatever scenario suits you, you can get the protein sequence, the primary sequence information in terms of amino acid. You have to get that. So let's take the sequence of BCL2 for example purpose. So this is UniProt site. So I'm taking the BCL sequence. Right now I'm taking human samples. I've already shown you these things in, I think, the UniProt video in detail. Right now, I'm just taking the sequences of BCL2. So let's take the plain sequences. So these are the protein sequences of your BCL2. In your cases, you can search for your protein either from UniProt or NCBI, and then just you have to provide these primary sequences in SOPMA platform for the prediction of your secondary structure. Okay, you just have to do in the default settings, submit, and in few seconds, few minutes, it's going to give you the result for the secondary structure prediction. So it's taking few minutes. Let it run, and meanwhile what we can do, we can put the samples in different platforms also. Like the second platform what we are going to use in this session is CyPred. So for in CyPred also, for secondary structure prediction we have to use CyPred. We are providing sequence information. So if you want high-end analysis also, in that case, you, depending on your needs, we have to select these things also. Like if you want to predict the transmembrane and helix prediction in the membrane also, then you have to select these part also. If your requirement is only for the secondary structure prediction, in this case you just have to click this one. Okay, so you have to provide the sequences, name of your protein. Although email is optional, but it's always best to provide the email also, because sometimes like it takes time to run, few minutes or sometime half an hour. depending on your samples and the size of samples. So it's always best to provide the links also. So you will be getting the result in the link form in your email. So we just have to provide this. If you have provided the samples in correct way, then it will start running. If the samples is having any error, then it will going to show you the error part also. So right now it's correct. So it is running. It will take some time. Okay. Meanwhile, we have got the result of SOPMA platform also. So you can see this page will look like this when you will get the result. So you have provided the sequence information about your protein, then within few minutes, we are going to get result like this. So they are mentioning the sequence length also. So you can see the length of your protein, and these are the alphabetical code of our peptide and amino acid. So here you can see for this is the protein start and this is the sequence. So number 10, 20, 30. So accordingly, we can get the idea. Here also you can see the color coding E, C, T. So what part is making coil, helix, turn, everything is given here in alphabetical code also, and also in the form of color coding. And the detail information is also present here. Like if you want to get the ideas about what kind of helix they are making, so alpha helix, phi helix, and these kind of things, how much in terms of presentations also, in terms of percentage also, they are giving you the idea. So helix, the type, the turns, the coil, where it is present, and how much the frequency of those things in the total protein length they are also providing. And here you can see the start of your protein in the color coding, what region contain what type of secondary structure. So it's also given in this form, your bar diagram. Okay. So for your thesis purpose, you can get these information from here. So either use SOPMA platform, they will also give you the detail about the secondary structures. CyPred result you have already seen. The third platform you can use is JPred. So this is the result of CyPred, what we have got. So after providing the sequences of your protein samples, something like this you are going to get. So you will get these things. The information they are going to provide you like sequence information and what kind of helix, turn, and secondary structures this protein is going to make in terms of prediction. So you can see this color coding here also, and what part of this protein is going to make strands, helix, coil. And in this way, we will get an idea about this protein. And depending on that, you can have an idea about their functionality also, the probable functionality and their things like in what way this protein is going to behave. So here you can see like helix is present here. In similar fashion, right now we haven't selected for MS, but if you want to get the ideas about the polarity, which part of this protein is polar, non-polar, which part is hydrophobic. So these kind of information is very relevant when you are performing biochemistry experiment or interaction studies. So you can get the ideas about your protein in terms of properties, what kind of property it could have. So you can get these ideas. If you want to download these things, you can very easily download here. If you want detailed information, you just have to click here and you can scroll here. So once you will scroll here, you will get the detailed information about this protein. So here they are providing you the alphabetical detail about your protein. So from start, what protein is present, and then they have alphabetical coding. So if this part of your protein is making coil, helix, so it's mentioned that it is CH. So coil, helix is present, and this is the cartoon representation of the same with the score values also. How much is the chance of these things, like in terms of probability? So you can see in the reagent section, the confidence of the predictions also, and what these color is representing. And for your thesis purpose, report purpose, or publication purpose, if you want to download, you can download these pictures also. Okay. So once you will, in the default setting, if you will just ask for secondary structure prediction, in that case, what we have shown you, you will get the secondary structure detail and the type of amino acid which is present. But if you want membrane information in terms of helix, what it is present, then in that case, you have to run memstat also. Just you have to click there in the settings while providing the input, and you just have to provide if it is present, the membrane helix are present. So in this result, you are going to get this result also. And here you will get the detailed ideas, like what, like if you will click here, show memstat, like when we haven't clicked on the default setting, the memset calculations and prediction, we were not getting this result. But now they are showing you the memstat. If you will select and then predict, then they are going to give you the ideas about the helix which is present, which could be present in the membrane. So you can get this idea that membrane interactions, what kind of extracellular amino acid could be present or no, and accordingly you can get the ideas about, or you can predict the functionality based on that, because different kind of extracellular transmembrane has different functionality also. So you can get the ideas about transmembrane helix, membrane interaction, and we have all the coding for that. And you can see there are probable chances that these protein could be present, and here in this form also they have given the ideas. So what is the amino acid detail and what protein could be in the extracellular region? So you can get this idea from here also. So with color coding, with sequence information like 143 to 158, these could be present in the membrane, and what part could be in the extracellular part, what could be in the cytoplasmic part. So you can get the fair idea about the transmembrane helix and what protein are present there. So if your work revolved around membrane proteins, transmembrane proteins, and signaling related protein, in that case you have to do these kind of prediction to get the ideas what part of that protein is could have those kind of signaling or membrane related functionality. So depending on that, you have a lot of features in the default setting. Depending on your needs, you can do particular selections also, and you can get the detailed ideas about your protein in terms of structure things at two-dimensional level, and based on their types and structure, you can predict the functionality, which could be finally validated in the wet lab scenarios also and via experiments. Okay. Now, the third platform which you could use for the secondary structure prediction is your JPred. So there's sample sequence already present here. You can try it with these samples, or maybe if you want to perform your own sample analysis, then you have to provide the sample. So I'm not taking the samples, example samples. We are taking the same BCL2 what we have used for SOPMA and CyPred. So these sequences I'm providing here. You just have to go to make prediction, and you are going to get this kind of result. See, BCL2 protein is a known protein and many people are already working in this sector. So a lot of information is already present about this BCL2 protein. So in that case, we are already getting a lot of tertiary structure information and PDB informations. So this means that if we are going to click here, we are going to get a significant amount of data, both in terms of secondary structure data, tertiary structure data, and most of the time validated NMR, crystallography, and homology modeling based data. But suppose in your cases, if you are handling de novo new samples, so in that case, we won't get this kind of result, because these PDB files we are going to get when someone has already solved the structure, someone has done the tertiary level structures. And if we have the primary data, then only we are going to get the secondary, tertiary, and then higher quaternary structures. So if we are getting this PDB files, it means we will get here tertiary structures also and secondary structures also. But if you are doing, if you are not having very known protein, and if you are handling de novo new protein, in that case you have to click continue button, and then based on your sequence, JPred is going to give you the ideas about your secondary structures. But in that case, this will be the predictive structure made by the computer. But here, because already structures are solved at the tertiary structure and higher structure level, so we are going to get much better and accurate structure in these sections. Okay, so either try SOPMA platform, CyPred, or if you are handling new sample, then you just have to click to the continue button, and based on the sequence they will give you the predicted secondary structure. But right now we are getting the secondary structures. You just have to look for the scores, like in the blast E value should be 0 or it should be more in minus. So every platform has some score values to get the ideas which samples are good or which are not relevant. So we have to get the ideas about the E-score value. So E-score looks decent and nice here. So name of protein is relevant. And as I already mentioned in UniProt tutorials also, like your protein is a macromolecule. They have different chains, and maybe in similar protein only, like suppose you are also working on BCL2, I am also working on BCL2, but your project could be on chain A, my could be on chain A, and your could be on chain B. So on same protein, different people can work on different project. So if you have specific project related to your protein, in that case you could click on that part of PDB. Other than that, the chances are, like based on the score values and your PDB, we have to analyze few samples. So let's take this chain A. See, this is a EMBL based tool and resource. So we are not going to give you the lot of information, the detailed information about JPred, because here a lot of information is present. Right now, our focus is on secondary structure. So I'm giving you the idea. Once you will click the PDB ID, you have from where you can get the secondary structure information, because I will make a separate tutorial, because here you can get a lot of different information in terms of Ramachandran plot, your secondary structure, the ligand interaction, the structure, pocket details, its hydrophobicity and all. So in one tutorial, I cannot teach you here everything, but this is really very useful platform. So you people who especially who are working on protein biochemistry, antigen, antibodies, your biotechnology related things, they can really explore this page. So right now our aim is to get the idea about the secondary structure, and for that purpose we have to go to this part. Okay, so here you can see that we have provided the protein sequence is also present with the secondary structure detail also, like how this protein is looking like, where is the helix, where is the coil, and all these things. Okay, so you can get this idea, secondary structure ideas from here, and from the main page you can get the tertiary structures and their PDB also, and you can download those things. But right now for secondary structures you can get it from here. So either you can use SOPMA platform, CyPred, or JPred for getting the secondary structure prediction of your proteins. I believe you have liked the session and found this video tutorial insightful. Don't forget to like and subscribe the channel, and do share these informations and tutorial among your scientific endeavor. Thank you so much.